postdoctoral fellow and later as a scientist. Her love for academia and curiosity to explore new ideas led her to join academics in 1993, when she took up position as an assistant professor at the Department of Molecular Reproduction, Development and Genetics at IISc Bangalore, where she continues to serve as a professor. She is also co-chair of the Center for Biosystems Science and Engineering, of which she was one of the founder co-chairs. In addition, she also holds the position of an adjunct professor as Faculty of Medicine and Dentistry, University of Bergen, Norway. She has to her credit numerous fellowships and awards, including the Fulbright Nehru Senior Research Fellowship and J.C. Bosch Fellowship. She was awarded the Central Drug Research Institute Award for Excellence in Drug Research and also the Y.T. Tathachari Award, to name a few. Her initiatives to improve translational research in India earned her the Margdarshi Fellowship from the Wellcome Trust TBT India Alliance. She was also a recipient of the Bill and Melinda Gates Grand Challenges Exploration Grant for her proposed work on a small animal model of ETEC-mediated diarrhea, which I think is what she's going to talk to us about now. She has more than 100 publications to her credit and is a member of various professional bodies like Indian National Science Academy, the Indian Academy of Sciences, Guha Research Council, and Third World Organization for Women in Science. Dr. Visweswariah primarily works in the area of signal transduction, cell biology, protein structure, and function. Her research group investigates molecular mechanisms of signaling by the receptor guanylyl cyclases, phosphodiesterases, and novel nucleotide cyclases in bacteria. Today, Dr. Visweswariah will discuss the work carried out in her laboratory on GCC signaling and a hyperactive GCC expressing transgenic mouse generated in the lab, which can serve as a model for chronic diarrheal diseases. So thank you, Sandhya, for being here with us, and it's great to have you. So over to you, you can start.
Thank you very much, Shailaja. And it's always been such a great pleasure to meet people in THSTI. I visited on a couple of occasions and been involved in other roles as far as THSTI is concerned. And it would have given me great pleasure to have been able to make the visit personally to deliver this talk. But the circumstances are such that I need to stay back in Bangalore. Now, what I'm going to talk to you today about is a story, one of the many stories that are ongoing in the laboratory. And I think this is an interesting story because it shows the way that perhaps someone trained as a biochemist and a chemist can move into thinking about translation, specifically gut physiology, and the way the gut responds to various environmental stimuli. Now, it was Hippocrates who said many, many years ago that all disease begins in the gut. And I think as we've gone on over the last decade, for sure, we've moved on from thinking about the gut as being purely an organ for digestion, but also one that's extremely important for fluid and ion secretion. I'll talk a little bit about diarrhea, which is one of the major diseases that afflict humankind all over the world. We've also become acutely aware of the fact that the gut is a rapidly proliferating tissue, and the entire intestinal epithelium regenerates itself once in 4 days, 4 to 5 days. Therefore, the whole process of proliferation needs to be very tightly controlled as far as the intestinal cells are concerned. And of course, we seem to have an overload of information right now on the role of the microbiome. The microbiome are the organisms that live within the gut, and we're now becoming more and more aware about how critical they are in impinging on very specific biological phenomena at the whole animal level. Now, the intestine is somewhat of a complex structure, and when I talk about it, I'm going to start right at the base of what are called crypts of the intestine. These harbor what are called intestinal epithelial stem cells, or IECs. Now, these IESCs express LGR5 GFP. So this is a stem cell marker. And these stem cells continuously divide and result in forming mature and differentiated epithelial cells that line the intestine. Important in the functioning of these stem cells are cells called Paneth cells, which lie at the crypts of the small intestine. These secrete antimicrobial peptides, which are very important in controlling the microflora that naturally inhibit the gut. The intestine contains goblet cells, which secrete mucus, which prevents the bacteria that are in the lumen from direct contact with the intestinal epithelial barrier. There are enteroendocrine cells, which secrete a number of gastrointestinal peptide hormones, including ones that I'll be talking about today. Now, of course, this tissue is also talking to the immune system, which is present in the lamina propria, as well as intestinal epithelial cells, immune epithelial cells. And all of these somehow bring about a very holistic functioning of this entire complexity. The immune cells regulate the delivery of luminal antigens and bacteria to the lamina propria resident antigen-presenting cells. The commensal microorganisms secrete many products which are beneficial in repair of the intestinal cells along the gut. And of course, the IECs, or the intestinal epithelial cells, possess mechanisms to allow for tolerance so that we don't elicit a very strong immune response to the normal gut flora that are present in the intestine. Now, I don't think diarrheal disease needs any introduction, especially to people in this audience. And it's one of the major causes of illness and death, especially among young children, less than 5 years of old, less than 5 years of age, in low-income countries such as India and sub-Saharan Africa. And unfortunately, almost 1 in 10 child deaths result from diarrheal disease during the first few years of life. And there are almost 1 million fatalities worldwide every year. And we have certain strains of E. coli, which are called enterotoxigenic E. coli, that harbor plasmids that produce a bacterial peptide toxin called the stable toxin. Now, many years ago, these stable toxins were purified. These are secreted from these bacteria, which is kind of unusual because they're synthesized as pre-pro-peptides by these bugs and actually are one of the very few examples of peptide secretion by bacterial cells. What is characteristic of these peptides, as you can see, and the sequence that I've shown you here are the mature peptides secreted into the medium. These peptides contain 6 cysteine residues, and these 6 cysteine residues are disulfide bonded in a very precise manner. Now, it is believed that it is this disulfide bonding that provides the heat stability of these peptides, and that is why it's called the heat-stable toxin. And even cooking food, in many cases, does not destroy this particular toxin if the E. coli are present in what you ingest. Somewhere in the 70s, it became apparent that these peptides elicited their action in the gut by increasing cyclic GMP levels. Now, this was a Nature paper which appeared in 1978 from the laboratory of Guerrant. This is Farhad Murad, who you would know as having got the Nobel Prize for his work on nitric oxide and cyclic GMP signaling mediated by nitric oxide. But it took almost another 20-25 years before the receptor for this particular toxin was characterized. This receptor is called guanyl cyclase C, or receptor guanyl cyclase C. C because it was the 3rd member of the family of receptor guanyl cyclases that were initially characterized by David Garber's group. What I'm showing you is a cartoon representation of the receptor. And it's important for all of us to realize that there is absolutely no structural information available on any domain of this receptor to date. The extracellular region of this receptor binds the heat-stable toxins and endogenous gastrointestinal peptides called guanylin and uroguanylin. Now, these peptides are secreted by the intestine and are believed to act in regulating GCC in terms of cyclic GMP production without any infection. I'll talk a little bit about linaclotide later. Binding of these toxins or the peptides to the extracellular domain elicits a profound conformational change through the intracellular domain, resulting in cyclic GMP production. In between the extracellular domain and the guanyl cyclase domain is a region which has similarity to protein kinases and is called the protein kinase homology domain, which is connected to the guanyl cyclase domain by a linker. Now, we've looked at almost every aspect of this particular receptor in some of the earlier years of our laboratory. We've made a series of mutations along this receptor, looked at the way different domains regulate the catalytic activity of the receptor following ligand binding. But I will not dwell too much on that at this point. Now, where is GCC expressed? Now, fortunately, in recent years, we've had single-cell RNA seq analysis of every type of cell from the intestine. And what you can see here is that this receptor is expressed in almost every cell type in the gut, including stem cells and their progenitors, goblet cells, Paneth cells, as well as enterocytes as well. How does GCC bring about its signaling action? Binding of ligands to GCC produces cyclic GMP intracellularly. Cyclic GMP now binds to the cyclic GMP-dependent protein kinase, PKG2. PKG2 phosphorylates two main targets in the intestinal epithelial cell. One is the cystic fibrosis transmembrane conductance regulator, or CFTR, and the other is a sodium hydrogen exchanger, NHE3. Phosphorylation of CFTR results in increased chloride efflux by the chloride channel, which is CFTR. NHE3, however, on phosphorylation by PKG2 is inhibited. Normally, this channel allows the import of sodium ions into the intestinal cell, but when it is phosphorylated by PKG2, sodium ions accumulate in the intestinal lumen. Now, the accumulation of sodium in the gut, along with chloride, results in an osmotic imbalance, and therefore water rushes out of the intestinal cell. And this is essentially the diarrhea that one experiences when these heat-stable enterotoxins bind to the receptor. Now, these toxins are superagonists of GCC, and it is believed that the endogenous peptides, guanylin and uroguanylin, which have a much lower affinity to the receptor than the ST peptides, elicit a cyclic G production level, which is just what is required to allow the fluid secretion to lubricate the gut, which is required for the passage of food, etc. So this whole signaling system was what was understood. We and others have extended some of these observations to show that GCC can also regulate cell cycle progression in colon cancer cells. We've also shown that the absence of GCC in GCC knockout mice can modulate infection by Salmonella typhimurium and Citrobacter rodentium. Again, something that may be a focus of another discussion on some other day. And of course, I can't forget PDE5. PDE5 is a cyclic GMP-specific phosphodiesterase, which is the target of sildenafil citrate or Viagra. And this is actually the enzyme that Shailaja spent a lot of time thinking about when she was doing her PhD. Now, around a decade ago, I was approached by clinicians from the Haukeland Hospital in Bergen in Norway. Now, they told me about a family of about 31 members, and many of these members of this family reported constitutive diarrhea, very frequent diarrhea on a daily basis. Some of them had as many as 20 stool passages per day, but on average about 3 to 4 stools per day, which was of loose consistency. Meteorism was also associated with this, with some members of this family, and abdominal pain. Now you can see when you look at this particular chart that the mutation appears to be an autosomal dominant mutation, and it does not appear to be sex-linked. Many of these patients suffered throughout their lives, and many of them had serious complications. Some of them had to undergo surgery. They had to undergo laparotomy to reduce regions of ileal inflammation. In many cases, there was twisting of the intestine and adhesion of the intestine to the peritoneal wall. So it wasn't just a simple case of diarrhea, but actually many symptoms which resembled inflammatory bowel disease and Crohn's disease. And indeed, many of these patients were diagnosed with Crohn's disease during their lives. Some of these are very young children. Some of them have grown to quite an old age of about 80 plus. Now, these clinicians, realizing that they had an interesting genetic association here, went on to try to identify where the locus of the mutation lay. And they did this through classical linkage analysis and identified that the region of chromosome P12.3, where this mutation lay, harbored a number of genes. And when they looked at the genes in this particular locus, they saw that GCC, or the gene name in, is GCC2C, was located in this region. Now, when they looked at all the other genes here, they did not find any direct association with diarrhea. So they decided that let's see whether there are mutations in the GCC gene in these patients. And what they found was that in all affected individuals, there was a single point mutation where an amino acid serine at position 840 was converted to isoleucine. So it's an S840I mutation. Now, this mutation lay in the guanylyl cyclase domain of this receptor. It's at this point that they wrote to me because they had come across our work where we had made a series of mutations in GCC and had studied the regulatory properties of those mutations. And they wrote to me asking whether we would be interested in characterizing this particular mutation. We, of course, readily agreed. And as you might expect, based on the phenotypes of the patients, this mutation was an activating mutation. Now, a couple of important things to note about this particular data here. These are wild type and mutant receptors expressed in HK293 cells, stimulated with the ligand and cyclic GMP measured. Now, this is a fold stimulation that you see with the wild type receptor. But in the mutant receptor, you can see that the levels of cyclic GMP produced are almost 5 to 10-fold higher than what you see with the wild type receptor. Another important point to note is that basal levels of cyclic GMP, that is the levels of cyclic GMP in the absence of ligand, were not changed. Now, in the gut of patients, this hyperactive receptor would have responded to the endogenous peptides guanylin and uroguanylin, presumably not the ST peptide, because it would be unlikely that these individuals in Norway were affected by E. coli-mediated diarrhea of this child soon after delivery. You're able to see this distended abdomen even in utero in scans. And when we brought in this mutation in the receptor, you could see that the activation by ligand was much higher than of the wild type. And importantly, even the basal activity of this receptor was significantly higher. So even in the absence of ligand, there was an elevated level, or there's likely to have been an elevated level of cyclic GMP in the gut. Now, many of these children are unable to retain any food orally in the first few years of life, and they need to be given total parenteral nutrition in order for them to survive. So the effect of this mutation is extremely profound as far as the intestine is concerned. This is another child. Her father wrote to me from Australia about his daughter and how when they did exome sequencing to see what her problems were, because she was also experiencing diarrhea, etc., there was a new mutation again, which again we characterized, and we found that it was a hyperactive mutation. So what we concluded from all of these studies, which have been reported everywhere, is that elevated cyclic GMP levels cause severe gut disease. Now, a number of mutations in GCC have been reported, and I'm only just cataloging them to you right now to highlight only a few things. There are recessive mutations, and these mutations need to be in a homozygous state to be able to be visible at the phenotypic level in patients. These are inactivating mutations, and they cause something called meconium ileus, which is the inability of a newborn child to pass the first stool. But somehow some of these recessive mutations also result in disease even later in age. Mutations have been reported in the extracellular domain. In the kinase homology domain, we long ago showed that the kinase homology domain was critical for ligand-mediated activation. Number of mutations have been reported there as well. Now, the linker region, again, this is a region where we have worked for many years and looked at the role of the linker in regulating guanylyl cyclase activity. Very interestingly, 2 of these mutations that were found in patients were exactly the same mutations we had made more than a decade earlier, purely to understand the alpha helical region that the linker region adopted. You can see that dominant mutations are, activating mutations, are dominant, and they're found in a heterozygous state, whereas, as I said, the inactivating mutations express a phenotype only under recessive conditions. There are mutations in the guanylyl cyclase domain. This is the S840I that we've spoken about, and all of this has been cataloged in a recent review. As a result of our work, mutations in GUCY2C now are recognized as being a contributing factor to early onset IBD. Now, this is a study of about 1,000 pediatric cases of inflammatory bowel disease in a single hospital, and that's the Hospital for Sick Children or Sick Kids in Toronto in Canada. And of the 1,000 children that they characterized, many of them had known monogenic causes of IBD, and GCC is now considered as one of the monogenic causes of IBD in these patients. Around 6% of these kids had mutations in GCC, and 2 of them have not been characterized earlier, and we're in the process of doing that in the laboratory right now. As you'd expect, early onset IBD is expected to be caused by genetic factors rather than the environment. And there have been more than 200 loci that have been associated with IBD through GWAS studies. But actually, they explain really only 14% of IBD and even fewer percent of Crohn's disease patients, clearly indicating that there are a number of other variants that are important in bringing about this disease. Now, one of the factors that struck us as we were trying to understand this whole phenomenon was that elevated levels of cyclic GMP not only caused diarrhea in humans, but also caused severe inflammation. And there was so far no reported link between the role of cyclic GMP and inflammation in the gut. Now, this inflammation is also seen in young children who are affected by ETEC or ST-mediated diarrheas. With repeated episodes of diarrhea, they do also experience inflammatory problems in the gastrointestinal tract. So this prompted us to try to think about developing a mouse model where we could introduce an activating mutation in these mice and characterize this mouse model at a level of detail that obviously would be extremely difficult to do in the human patients as we have them. Now, over the next few slides, I'm going to summarize very briefly this paper that we published last year, where we talk about gut-associated cyclic GMP mediating colitis and dysbiosis in a novel mouse model harboring an activating mutation in GCC. Now, before we began this work, it was important for us to show that the equivalent mutation of the S840I seen in humans had similar biochemical properties in the mouse receptor. So the position of the mouse receptor is S839I, and this is because of a single amino acid missing in the linker region in the mouse receptor. So we generated the mutant, we cloned the mouse receptor, generated the mutant receptor, and tested for its expression in HEK cells, and also looked at the levels of cyclic GMP produced by the wild type and mutant receptor in the presence of ST, as well as in uroguanilin. And you can see that at all concentrations of the peptide, the level of cyclic GMP produced was higher in the mutant receptor than in the wild type receptor. Though more interestingly, you can see that the increase in cyclic GMP production seemed to be far more evident with uroguanilin as the stimulating peptide. Interestingly, as we saw in the human receptor, the EC50, or the half maximal dose required to elicit a full response, was lower in the mutant receptor. And therefore, with the knowledge that if we were to introduce this mutation in mouse GCC, we are likely to mimic the phenotypes seen in humans, we generated this mouse. Now, this mouse was generated before the days of CRISPR-Cas. So it took almost 3 to 4 years before we were able to receive this mouse in our colony for various reasons, not purely scientific. And the mouse was generated by Taconic in Denmark. We, of course, began initially by characterizing this mouse model for expression of GCC, obviously, as well as the 2 endogenous ligands, which we expected to activate the receptor in the gut of these mice. We could see at the level of Western blot analysis that levels of GCC were similar, in agreement with the quantitative real-time PCR. And therefore, we felt that we had a model where expression levels of the receptor were not significantly different in the wild type and the mutant. We then went on to actually measure cyclic GMP levels in the gut of these mice. And this is by preparing epithelial cells from these mice and actually measuring cyclic GMP using ELISA. And it was heartening for us to see that the levels of cyclic GMP found in the gut was significantly higher in the mutant mouse, which is what we would have hoped for, and we indeed saw, whether it's in the ileum or in the colon. Now, mice don't get diarrhea the way humans do. At least C57 black 6, in which this model was generated, do not. And therefore, you require slightly fancy methods to check whether they have any kind of aberration in terms of bowel function. And therefore, what we do is we measure the number of fecal pellets that a number of mice pass in 10 minutes. And you can see that there was an indication of a higher number of fecal pellets passed by the mutant mice. We can collect these fecal pellets, lyophilize them, measure the weight of the wet fecal pellet and the residue left after lyophilization, from which we can calculate the percentage of water content present in the feces of these mice. And you can see that that was also significantly increased, mimicking the kind of watery stools that patients may experience. As I said, the diarrheal phenomenon is much, much milder than you see in humans. However, there are certain times where my students come back and tell me that they felt that the fecal pellet was a little softer and a little more squishy from these mutant mice. Now, if you remember, one of the downstream targets of signaling via GCC was the sodium hydrogen exchanger NHE3. So we decided to look at all the signaling components downstream of GCC, which cause diarrhea, whether it's cyclic GMP dependent protein kinase, PRKJ2 is the name of the gene, CFTR, and NHE3 or SLC9A3, as it's called at the genetic level. There were no significant changes in levels of PRKG2 and CFTR, but very interestingly, we saw that NHE3 was significantly downregulated at the transcript level, as well as at the Western blot level. Now, this is interesting because GCC, as I told you, inhibits NHE3 through phosphorylation by PKG2. What we're seeing here is actually a further downregulation at the transcriptional level, presumably by the elevated levels of cyclic GMP in the gut. Now, this is something that we may pursue and look at a little more in detail in the years to come as to how cyclic GMP could possibly be regulating the transcription of NHE3. Now, if NHE3 levels are significantly down in the gut, this would mean that there would be an accumulation of sodium in the gut lumen, which would now pass out in the feces. And so you can actually measure sodium in the fecal samples, and you could see that indeed levels were increased in the S839I mice. Now, as I told you, one of the other interests was to be able to look at the inflammation that's seen in humans and try to find out how models behave in response to elevated levels of cyclic GMP in the gut. Now, one of the standard models to look at colitis or induced colitis in mice is by applying DSS or providing DSS in the drinking water to these mice. So we induced colitis in both wild type and mutant mice. Note that we co-housed these mice, and this was mainly to avoid any environmental factors that are known to affect the degree or extent of colitis in animals. These mice were fed either water or 2.5% DSS for some days and then put back on water. And over the course of the experiment, we measured the weight, and you can see that the mutant mice lost a significantly higher degree of weight, or more weight loss was seen in the mutant mice. Now, one needs to ethically sacrifice mice once they lose weight beyond 20 to 25%, which is why the experiment ends here. You can also look at the disease index in these mice, and this is by looking at things like blood, rectal bleeding, a little bit of water and a kind of mucus in the husk. And so all of these parameters also indicate that the mutant mice were more susceptible to DSS-induced colitis. This was also visible when you looked at the histological sections. Colitis is associated with severe damage to the colonic epithelia. And you can see that a very large number of immune cells had infiltrated into the mutant colon on DSS treatment. Please note that the colon looks very normal in the wild type and S8 mice in the absence of DSS. And this again could be graded, and you can see that there was severe damage or more severe damage in the S8 mice. Now, why is this happening? Obviously now, one of the things that we're always very interested in is thinking about the microbiome. And so we looked at the fecal microbiome in these mice, And you can see that there was a significant difference between the fecal microbiome of wild type and S8 mice. We went on to look a little bit more closer at the genera and the families that were affected. And what one sees here is that many of the good bacteria, such as lactobacillus, were all lowered in the mutant mice. And in contrast, bacteria which were known to be harmful, like Paraprevotella or Mucocespirilum, were elevated. Now, very interestingly, these are some of the similar changes that are seen in patients with Crohn's and ulcerative colitis. So the environment of the gut in our mouse model seems to mimic that is seen in human patients. Using a rather sophisticated tool called Epithelial, which actually looks at the genomes of the various organisms that are seen in a microbiome analysis, looks at what they could be doing and therefore predicting what pathways they could be regulating, we indeed found that at least significantly the microbiome in SA39I mice could induce inflammatory signaling, such as IL-17, Th17 cell differentiation, and even nod-like receptor signaling. Now, all of these pathways are known to be aberrant in Crohn's disease and IBD patients. So I think what we've been able to show here is a clear link between cyclic GMP levels and the regulation of the microbiome, which could be contributing to the inflammation. Now, we decided to probe this a little further by doing bulk Rna Seq of colonic tissue. And when we analyzed our Rna Seq data through Ingenuity pathway analysis, we found a very strong signature for STAT1-mediated gene regulation. A number of STAT1 targets were upregulated in the colon of these mice, significantly with FDR value that was acceptable. So we went on to validate this at the level of qRT-PCR and show that indeed genes like interferon gamma, IFIT1, IRF7, many of these interferon-stimulated genes were upregulated in the colon of these mice, as was STAT1. This was confirmed at the level of Western blotting, where we showed that there were changes in phosphoserine 727 phosphorylation of STAT1, and indeed, even total STAT1 levels were higher in the gut of these mice. Tyrosine phosphorylation was also significantly increased, and we also saw increased levels of some of the ISGs. Probing further the overall RNA-seq, and again, looking in Ingenuity pathway analysis, we compared the gene expression profile with that seen in mice who had been treated with DSS. Now remember, our RNA-seq was done from mice as they were without DSS treatment. But you can see that there was an inflammatory gene signature already present in these mice, which was induced on DSS in normal control mice. Interestingly, we also found a parallel gene signature in patients with colitis. And these are what are called upstream regulators. Now, these are actually genes which regulate a number of other genes downstream. And you can see that even the upstream regulators, such as STAT, etc., were also altered similarly in patients with colitis, as were in our SA39I mice. Now, this data is from upstream regulators from mice treated with DSS. This is actually a comparison of the expression levels of certain genes from human patients with colitis. So now this is the first model, which is the first preclinical model for colitis induced by activated GCC and by extension cyclic GMP. Dysbiosis is observed. Now, is this a consequence of altered iron and fluid secretion in the gut mediated by GCC? We think so. We now ask, what is the link between GCC, cyclic GMP, and inflammation? We're in a position to be able to probe this a little deeper using this mouse model. And one can ask whether the microbiome is a consequence or cause of inflammation. Now, what I'll show you is some more unpublished data from the same mouse model. The earlier studies that I spoke to you about focused on the colon and colitis changes. But as I mentioned to you, Crohn's disease is also one of the disorders seen in these patients. And Crohn's disease can affect many regions of the gut, but predominantly the small intestine. So we decided to look at what could be happening in the ileum in these transgenic mice. We first looked at overall histology, which was not dramatically different. But when we performed parietic acid shift staining, which specifically stains Paneth cells, which are found in the crypts, I hope it's obvious to you that you could see a significant reduction in the number of Paneth cells. Maybe not the number, but at least in terms of their ability to secrete the active peptides that they do. We went on to study this using transmission electron microscopy in collaboration with Dr. Annie Pulimud in Christian Medical College in Vellore. And again, looking closely at the Paneth cells, you can see the nice lysozyme-filled granules that are seen in wild-type mice, but seem to be far reduced and far less stained in SA39I mice. If we go back to our RNA Seq analysis, which we performed in the ileum, you could see that a number of Paneth cell-specific markers were downregulated, and importantly amongst them, the alpha defensins, which are known to be produced by Paneth cells to protect against microbial insult. So we now had evidence to suggest that hyperactive GCC in the ileum of these mice brings about changes in the Paneth cell. Now, Paneth cell function is regulated very precisely by autophagy. And so this throws open another very interesting avenue of research in future as to whether there is a link between cyclic GMP and autophagy, which is something that people haven't studied earlier. Now, when you look at the RNA Seq that we performed in the ileum, again, we found a very significant association with the genes that are misregulated in Crohn's disease patients. We validated many of the genes that were misregulated in the SA39I mice at the level of RT-qPCR, as well as at the level of Western blotting. And one thing that I think I'd like to highlight is the fact that at least as far as the ileum is concerned, the extent of activation of STAT1 seems to be far more dramatic than what we saw in the colon. And this was also evidenced by the levels of many of the interferon-stimulated genes. Is this only at the level of protein or at the level of RNA Seq or the level of transcripts? What about functional cytokine production in the gut of these mice? So here we prepare tissue extracts from the ileum and the colon, and we perform ELISA for various cytokines. And as you can see, many of the inflammatory cytokines, such as TNF-alpha, interferon gamma, IL-6, many of them are upregulated in the gut of these mice. Interestingly, IL-22, which is known to help in the repair of the intestinal epithelium, is really not much changed in the gut of wild-type and S839I mice. Many of these changes were also seen in the colon of these mice. So this is now actually measuring active cytokines, or perhaps the precursor cytokines in the tissues of these mice. We went on to do multicolored FACS analysis by isolating the intraepithelial lymphocytes, as well as the lamina propria cells, and also cells from the MLNs, the mesenteric lymph nodes, and the Peyer's patches in these mice. And what I'm showing you here is not a very complicated analysis, but a summary of what we saw. The levels of immune cells were increased in the blood and in the MLNs, as one might expect. We saw T helper cell levels significantly increased, and a reduction in some of these monocyte to macrophage, as well as other monocyte lineages being lower in the blood and in the MLN. In the Peyer's patches, the overall number of immune cells increased without a significant change in a particular subclass. So this could just mean that there is more recruitment of immune cells to the Peyer's patches because of the stimulus that they're receiving from the gut. Interestingly, there was no change in immune cells in our preliminary analysis in the lymphocytes that are found as intraepithelial lymphocytes or found in the lamina propria. There was no change in the cells that we measured. We could then ask us whether the functions of these cells are altered, and these are some of the questions that we will be addressing in future. Finally, everyone is excited about gut organoids and what they may do and how they may help us dissect out what are cell autonomous phenotypes that is mediated purely by the intestinal epithelial cell versus cell non-autonomous effects in the gut of these mice, which could be brought in by the lymphocytes and other cell types in the gut. And therefore, we set out to try to establish organoid cultures from the gut. We obtained these organoids initially from stem cell, cultured them in the lab. Now, these are the organoids which contain these LGR5 positive cells. We were able to culture them till they formed mature organoids, and we were able to freeze them. And of course, this work was largely the result of many years of work by Hans Clevers working in the Netherlands. We took these organoids and we treated them with buffer or nothing. We treated them with the stable toxin peptide or the uroguanilin peptide. And please watch these movies, which I hope play. Okay? You can see that in untreated organoids, there's even a suggestion that there is some proliferation over the 48 hours of imaging. I can show you again where you actually see them growing in size. Now, when you took organoids and you applied the stable toxin peptide, I hope you note the swelling of these organoids as fluid accumulates in the organoid lumen. Now, this is true when we added the stable toxin, as well as when we added the endogenous ligand uroguanilin. But you can see that the extent of swelling is somewhat less. We haven't quantitated any of this data. We propose to do that. But you can see that the swelling is significantly lower in the uroguanilin treated organoids. This black patch that you see at the end of the experiment are a large number of apoptotic cells which actually peel off here and get trapped within the lumen of the organoid. So now we have this very nice model system to be able to start looking at ways by which we can maybe inhibit this swelling brought about by ST. We can make organoids from both wild type and SA39I mice and look for inherent swelling seen in the SA39I mice, just purely because of hyperactive GCC. And we could try to reverse that swelling by using treatments known for the treatment of diarrhea, which could be efficacious in at least the clinical setting in years ahead. So where are we going to go with all of this? And these are mainly gut instincts, at least the gut instincts of my group and perhaps myself as well. What are the mechanisms by which we see panel cell defects? Is there a link between autophagy and cyclic GMP? Could we use organoids or these mouse models to investigate therapeutic approaches? Now, one of the most common therapeutic approaches that is used for the treatment of diarrhea is zinc. And zinc is known to at least alleviate quite a few of the diarrheal symptoms seen in children in countries like ours. What happens if you provide zinc to these mice? Now, preliminary experiments, I'm not showing them to you here, do suggest that the diarrheal phenotypes, such as increased fecal passage or increased bowel movement, as well as fecal water content, is significantly reduced. And most interestingly, we seem to see a recovery of panel cell function in the presence of zinc. So these are all preliminary results. We're going to try and see if we can push that a little further. Of course, you might ask me, are there no GCC inhibitors? Unfortunately not. There are no known antagonists of GCC. However, there is one compound which has been described, which appears to inhibit the catalytic activity of GCC, and that would be very useful to test in our organoid models, at least in the transgenic mouse. As I said, we will be developing organoids from these mice. Do we see increased swelling? We can actually look at the panel cells in these organoids directly. Are there changes inherent to the intestinal epithelial cell, which lead to dysfunction of panel cells? Or is there some kind of crosstalk that's happening, perhaps between the microbiome in the gut of these mice? And so that would be an interesting question to address. We have LGR5 GFP positive mice in the lab, and so we would hope to make crosses with these mice and our transgenic mouse to be able to actually visualize LGR5 GFP positive cells in the gut of these mice and look at stem cell function. Because as I told you, GCC is also expressed in stem cells. So is there any effect of this hyperactive GCC in the stem cell pool in the intestine of these mice? Obviously, there's nothing grossly wrong with these mice. So to a certain extent, these stem cells are functioning normally. But in the presence of a stressor, whether it's something in the diet, whether it's an infection, do we now start seeing aberrations as a consequence of hyperactive GCC expressed in these cells? And finally, at the level of translation, all of this has got me very interested in thinking about very early onset IBD in India, and whether we do indeed see mutations in GCC2C in Indian children. In fact, working with a few clinicians in Bangalore so far, they have provided us some evidence from their whole exome sequencing analysis that some of the children they've seen do indeed harbor variants in GCC. And now we ask the question whether those variants are significant in terms of regulating GCC activity. In a larger panel, maybe across 100 to 200 very early onset IBD or pediatric IBD cases in India, we plan to do whole exome sequencing and possibly develop an India-specific gene panel, which would be of use to pediatric gastroenterologists in the country, because they tell me that there are many variants that they've tested which are reported in the Caucasian population, which are not found in the Indian children that they see. And of course, ultimately, look and see whether all variants in GCC contribute to diarrhea and inflammation in the gut, or are there some variants which are not that important in terms of regulating GCC activity. Now, before I close, none of this would have been possible without a really exceptional group of students that I've had the great fortune to have in the lab. There have been more than 30 that have passed out of the lab with PhDs in various other areas of research, but I'm highlighting here the students that have really contributed to what I've spoken today. Dr. Najla Arshad was the one who characterized the S840I mutation, working with Norwegian clinicians. Insha was a master's student who characterized additional mutations, working with clinicians from Austria. Viswas was the first one to have the courage to even think about working with this mouse model. And he was ably assisted by Avipsa Bose and Yoshika Bopana, and much of the unpublished data that I've spoken to you about, and indeed, much of the published data that I've spoken to you about has come from the contribution of these two in the laboratory currently. Dr. Shashikaran did the entire microbiome analysis. I was fortunate to have her as a postdoctoral fellow in the lab. Dr. Sangeeta Banerjee also appeared in the lab at exactly the right time. She was a bioinformatician and therefore helped us significantly in many of the Rna Seq analysis. Dr. Idris Shah, Pooja were also there assisting all of this work over the course of the last so many years. And finally, one cannot thank enough the veterinarians who have been associated with the lab for the last so many years. Their presence is essential to maintain the colony and do all the complex breeding, because I haven't spoken to you about other mouse models that we have in the lab for GCC. We have knockout mice, we have another mutant, which was funded by the Bill and Melinda Gates Foundation. And so maintaining and breeding these mice, the LGR5 GFP positive mice, was really an effort. And I thank really both these two veterinarians for the exceptional work that they did. And Harini is still with us and hopefully will be for some more years to come. Now, funding, of course, none of this work would have been possible without significant funding. I was fortunate to get funding from the Bill and Melinda Gates Foundation, which assisted in generation of the transgenic mice. The Research Council of Norway also provided some support for the generation of the S840, the S839I mice. I have the collaborators that I'd like to mention. Dr. Torun Fiskerstrand, unfortunately, she passed away a few years ago, but she was the one who really drove the S840I project in Norway. Dr. Halvor Sommerfeld, I think many of you know, we've been collaborating with him and others in Norway on trying to develop a vaccine for stable toxin-mediated diarrhea. In the UK, I have a Royal Society grant for collaborative for professors, collaborative research grant with Professor Gad Frankel in Imperial College London. Dr. Avinash Shenoy was a former student in my laboratory who's also very interested in looking at inflammation in the gut. And both of them work at the MRC Center for Molecular Bacteriology and Infection. Thank you very much for your attention, and I'll be very happy to answer any questions that you might have.
Thank you very much, Dr. Visweswariah. I think this was fantastic talk, and you know, people sometimes think that you can just do it in a few years' time, but this, I think, is a lifelong work that you have presented in such a short time. And I think the beauty here is the originality and the novelty that you have brought into this kind of research, taking a very, very small molecule I think, which has such profound physiological function and also is a target for therapeutics, not only for diarrhea. And that's what my question would be, that do you think this is something which can be used for treating constipation also? I don't know whether there's a...
Excellent. Thank you very much. Thank you so much for this question. I did mention that I would mention linaclotide. Yeah. Actually, given the fact that these stable toxin peptides cause diarrhea, a number of companies in the US have developed agonists for GCC, which is nothing but the 13-mer core sequence, the cysteine-rich core sequence in these peptides. One of those drugs is called linaclotide. It's an FDA-approved drug specifically for the treatment of constipation. So you're turning on this pathway to relieve constipation. Thank you for the question, because I think I forgot to mention it. Yes, absolutely.
Very good. Very nice.
Nice talk, Sandhya. If I can ask a couple of questions.
Of course, Ramadeep. Thank you.
So what was the, like you mentioned that you would want to study autophagy, right? So was there any evidence from the Rna Seq data which makes you think that autophagy?
Not really. Not really. We haven't looked that carefully specifically for pathways, but I think many of these effects could be post-translational and post-transcriptional. We may or may not see a significant increase in autophagy genes, but more their regulation in terms of inducing autophagy. The only reason I picked that up was because there's a wealth of information on Paneth cell function and how the secretion of lysozyme and things like that from Paneth cells is mediated by autophagy. So it's a hypothesis. Do I have tangible proof for it? Not yet.
Yeah, my other question was the Western blots for STAT1 and other genes like TAP1, were they in basal condition or?
Yes, all basal.
They were
all basal. All basal. Of course, following DSS induction, the wild type mouse would also respond. I mean, we have done QRT-PCR with DSS treated mice. But at the end of the experiment, almost the wild type mice respond as much as the SA39 mice. So all the data that I've shown you with respect to these mice was just basal inflammation that is seen in these mice without any treatment.
And are these mice more susceptible to infection?
Very nice question. These are the kinds of things that we'd like to study in future. We've studied the role of GCC knockout mice, and they are more susceptible to Salmonella typhimurium infection, as well as Citrobacter rodentium infection, one an invasive pathogen, one an adherent pathogen. We are currently doing experiments with these transgenic mice and in these using infection models. Another important point that I didn't talk about is how GCC regulates cell cycle progression. And we've shown that colonic cells, their proliferation is controlled by activation of the receptor. So now, by any chance, are these mice more or less susceptible to carcinogens that are provided exogenously? So these are, again, some of the things that we would like to try with this mouse model in due course.
Yeah, I think I will stop here. There is a question from Shinjini. I think
Raman, in between, may I request ma'am to please stop sharing the slide so that we can see ma'am and we can see the person who is asking question also.
I'm not sharing. I'll stop sharing.
Slide.
Yes.
Oh, yeah, that's perfect.
Sorry.
This is what I was
Thank
asking.
Yes, thank you so much. Thank you.
Shinjini ma'am, please go ahead.
Shinjini, are you there?
I'm there. I think we can ask Dr. Gar's mic is, yeah. So, hi, Sandhya. Thank you so much for a very, very interesting talk, and especially being a pediatric diarrhea person for more than almost all my life, clinical life. So we've discussed this in the past also when we spoke about chronic diarrhea. And my question was that, you know, when we actually see pediatric diarrhea, we don't see these cases very commonly in India. So this is a rarer form of chronic diarrhea that we are seeing. And but we don't have a cause for a lot of our congenital diarrhea when we do see them, especially in tertiary hospitals like AIIMS, where I was. Yes. So when you say you've been partnering with the clinicians, what is the kind of, are they seeing it as congenital diarrhea? Are they seeing it as toddler diarrhea, or are they seeing it in older? Because if there is a predilection for an inflammatory bowel disease in them, what would be interesting to see what happens to them as they grow, if they survive that period at all. Our experience is that most of these congenital diarrheas, we don't have a treatment for them and they die. So, because our experience with enteral feeding and all isn't the greatest given the infective environment that we have around us. So any thoughts on that?
You're absolutely correct, Shinjini. Actually, when we first reported these mutations, I spoke to pediatricians in CMC Vellore and asked them, are you saying that you do not see children with congenital diarrhea? And he said, yes, we see them, but we lose them because we're not able to take care of them. The work that we plan to do with the clinicians is to look at early onset inflammatory bowel disease. It need not be congenital, but the age of onset in the cohort that we've decided to focus on is below 18 years. So the child may come very young, and some of the cases that the pediatricians do see are a couple of years old. Some of them come when they're 5 or 6 or 7, and some of them come even in their teens. So we're spreading it a little bit more, not just purely focusing on GCC-mediated diarrhea, but trying to get an idea of where we stand with mutations in early onset IBD in the pediatric population in India.
So maybe just a suggestion in order to study this, because this is an uncommon event given it would be nice if you do it like a case control study rather than rather than following a cohort, because then you'll be able to identify the associations better. Yeah.
So, if I can just clarify, maybe I wasn't clear, but what we plan to do is whole exome sequencing of 100 to 200 patients, but we'd like to do a trio analysis. So we would like to take the parents' samples as well. And so that may help us identify variants a little easier rather than just look at the patient samples. Yeah. So that's what the approach we're trying to do.
No, that'll be great. I'll come back with more questions. I'll let the others ask more. Sure. Otherwise, I'll connect with you on a one-on-one for this.
Sure.
Thank you.
Absolutely. Thank you. Thank you.
Raman, over to you.
Krish, do you want to go next?
Dr. Sandhya, this was an excellent talk. I think a lot of us dream in order to make sure that we can do this in a lifetime. Couple of questions. One is in terms of, given that the cyclic GMP accumulates so much, what happens to this imbalance of the GTP cyclic GMP?
We haven't measured GTP. The levels of cyclic GMP are much, much, much lower than normal cellular pools of GTP. So there's likely to be inherent control within the system. For example, the phosphodiesterases are still there, and they may be cleaving a large fraction of cyclic GMP off. But cyclic GMP is at picomolar and nanomolar levels, whereas GTP is usually in hundreds of micromolar or even millimolar levels. So I don't think it's changing the GTP pool too significantly. But we haven't measured it.
Okay. So the second question was in terms of you having challenges with respect to identifying the Indian neonates or the children or the adults with respect to the SNPs, specifically in this particular receptor. I was wondering, isn't it possible to go back to each of these individuals who have diarrhea, look at their cyclic GMP levels, right? And then go back and ask, what is the mutation you have?
Yeah. So the cyclic GMP levels that we've measured are from actually pieces of gut. Now, when we work with clinicians, if any of them are performing surgery on such children, we might be able to get a piece of the gut and measure cyclic G. Cyclic G would be secreted from the epithelial cell into the lumen, but then the dilution would be so huge that we would not be able to detect it. I would think. We haven't done it, but I don't think we'd be able to detect cyclic GMP, for example, in the fecal pellet. We haven't done it. It's an interesting thought. I might see if one student's interested in trying. But I don't think that it would be very easy for us to actually measure cyclic G in the gut. I mean, there are hundreds of things we can think of. We have beautiful fluorescent sensors for cyclic GMP now, which respond to levels of cyclic GMP. If we could generate a transgenic mouse, and such mice are there, where we could cross that mouse with our mouse, and we could actually look at elevated cyclic GMP levels in situ, that would be a dream, but I'm not sure whether we're going to be able to do that. So in humans, it's, I think, difficult. And that was a limitation in the initial study also. While they reported all these inflammatory characteristics in the family, the Norwegian family, they had no real idea as to why that was happening. They've performed a fecal microbiome analysis. They've seen some changes. They've done a bit of Rna Seq. They saw very few changes. We're actually seeing much more changes in our mouse model than they've reported in the human model.
Would that be possible to see in urine in kids?
So this brings us to another point. GCC is also expressed in the kidney. Now, this animal knock-in we have is a whole animal knock-in because we wanted to mimic exactly what was seen in human patients. So therefore, it's known that GCC knockout mice have problems in kidney function. And, so it's a completely different kind of physiology that one would need to think about. But it's a point well taken. We could measure cyclic G in the urine and see whether we see changes. It's a good point. I'll make a note of it. Because we can collect urine easily with our metabolic cages, and it might be something that we could try and look at.
Thank you very much.
Thank you so much.
Atul?
Is this me now, Raman?
After Atul, Anna. Yeah. After Atul. Atul? You have to unmute yourself. You have to unmute yourself. Atul, we can't hear you.
Yeah. Yes, now I'm unmuted.
Yeah.
Thank you, man. Thanks for such a wonderful talk. My question is from the microbiome part of your study. As we can see that you see enhanced level of desulfovibrio, which is known to produce hydrogen sulfide, and hydrogen sulfide is linked to alter the redox state of guanylyl cyclase. It reduces the heme group of guanylyl cyclase, and also it is being implicated in intestinal inflammation in colitis model, murine colitis model. So I just wonder if you have checked the level of H2S or try to target H2S synthesis pathways to see the effect on colitis.
No, we haven't. Excellent thought. And perhaps I'll contact you and find out ways by which we could try and check whether inflammation is reduced in the presence of an H2S production inhibitor. The H2S has been shown to affect the soluble guanylyl cyclases. Now, I didn't go into these enzymes, but these are the ones with the heme center. And the receptor that we work with is not part of that family. So while it's a guanylyl cyclase, it's completely different. So, but I take a point in terms of H2S levels being elevated in CD and IBD patients. And this is something, again, that we could measure. Maybe we could measure it. Yep. Thank you.
Could it be used as a biomarker for inflammation in colitis model?
I mean, if it could, I'm sure people would have tried. I think because, I think the clinical picture of inflammation is so clear because, you take biopsies and you can actually see the way the gut has been affected, that I'm not sure that you need these kinds of screens. I don't know, I'm not a clinician, but I could check with the clinician, but I think for them, the inflammation is very obvious.
It's difficult to assess H2S being a gaseous product. So maybe in urine or some.
Yes.
Matter if you can quantify. Thanks, ma'am.
Thank you.
Anna, do you want to go next?
Yeah. Hi, Sandhya. This is Anna.
Hi, Anna.
Yeah. I just.
Anna, George. Okay. Yeah, sure. Yeah.
Yeah. Given that disease is due to a single mutation, is anybody trying gene therapy approaches? I mean, we can do all the biochemistry and all the genetics and all the microbiome and all the transcriptome, but ultimately, this is a disease in a minority of people, and it's identified as a single mutation. Is anybody working on this?
No, not at this point. You know, I've been fortunate that people haven't been so interested in this receptor for many years. It's only now that people are waking up after all the work that we've done and others have done, of course. So nobody is thinking about this. You know, one could think about this as prenatal screening as well, because as Shinjini mentioned, some of these children are really poorly, and the clinicians tell me that these kids are really quite sick. So if there's any sort of preclinical screening, but you know, those mutations in these young children are all de novo, they're germline mutations. And there would be no reason to test for the child to have such a mutation routinely, because the parents are fine. So I don't think anyone is trying this. And you know, you'd have to correct the mutation in the stem cell in the gut. It's not easy to do.
I understand all that, but since you have a mouse model, you can see whether you can correct it genetically.
We can, if we see phenotypes with the organoids, we could try to correct it in the organoid model,
right,
and as proof of principle, without having to do it in the mouse, but definitely as proof of principle. And I would think if people get interested, they are likely to do that, even with human organoids prepared from patients or humans. They would try to correct this defect, which people are doing with CFTR, for example. But yeah, nobody, to my knowledge, at least nobody has written to me saying that they're interested in doing that.
Right. Thank you.
Thank you, Anna.
Guru?
Hi, Sandhya, Guru here.
Hi, Guru.
Sandhya, so the effect of GCC, would you attribute everything to increase in cyclic GMP levels?
Not at all. No. I think it's a driver. It's a major driving force. For example, you know, this swelling in the organoids that we see, we can mimic that using a cell permeable analog of cyclic G. So that aspect of the pathway is through GCC. But subsequent to that increased fluid and ion secretion in the lumen of the gut, is that affecting the microbiome, which now comes back and affects the intestinal epithelial cells or the IELs and LPs? You know, these are all it's a consequence of cyclic G, but is everything driven through cyclic G? I think cyclic G has multiple effects which now go on. So it's a kind of broader picture than everything being cyclic G. There's no evidence to suggest that GCC signals independent of cyclic G through protein-protein interactions or anything like that. I think people still think about GCC as a cyclic G producer. And therefore, anything that happens is happening downstream of cyclic G.
Okay. So what is the connection between cyclic GMP and inflammation? Is anything known?
No, that's exactly why we're exploring it. What we have is evidence to suggest that there is an inflammatory signal in these mice. Now, the issue is that the microbiome is also changed, right? So now, is that inflammation a consequence of the altered microbiome? Or is it directly a consequence of cyclic G? So now in organoid models, for example, if we now add cyclic GMP, can we elicit a STAT1 signature? Because in organoids, we don't have any microbiome there, right? So these are some of the questions that we would ask at this point. For example, the activation of STAT1, is it because of the whole gut environment, or is it cell autonomous? Is it completely dictated by the intestinal epithelial cell and cyclic G? Things we'll look at.
Yeah, the signature you showed in your Rna Seq, I see predominant antiviral response. So I'm even curious to see whether they would be resistant to viral infections when there is activation of GCC.
Take these mice and do what you want, Guru. I have absolutely no problem. Actually, the minute I saw the ISG signature, and when I started thinking about where this whole problem started with those Norwegians, I really fancied that there was something happening in terms of norovirus infection, right? So is there some susceptibility to viral infection in the gut? And one of the things that we'd like to do, what we've looked at so far, is the bacterial microbiome. I mean, I would like to do metagenomics. And let's look at phages and the viral signatures in these gut of these mice. And I wouldn't be surprised if actually there are some viruses that seem to be happier in the mutant mouse and thereby generating. Yeah,
Okay.
totally, absolutely.
Thank you. Thank you.
Suchitra?
Hi, Sandhya. Suchitra here.
Hi, Suchitra.
Hi. I have a very quick question in terms of basic biology. You probably mentioned it and I missed it, but does cyclic GMP affect cellular proliferation?
Yes, we've shown that it inhibits the cell cycle in the G1 phase, but this is in cancer cells. And all the studies so far have been done with colorectal cancer cells. There are very few normal intestinal cell models available. And those that do not express GCC. So we'd have to put GCC in there or elevate cyclic G in another way.
So yes, I was just thinking that there's a very strong likelihood that, from using your LGR5 GFP model, probably there might be, I'm thinking that turnover of stem cells is definitely going to be affected. Yes. You're definitely going to see that effect.
Yeah. Exactly. And that's really a dream that hopefully we'll achieve once we cross these mice and get enough of them to make organoids, etc.
Yeah. Thank you.
Yeah. Thank you.
Nishit?
Hi, Sandhya. Very excellent talk.
Thank you.
Nice to see you here. So
nice
I was wondering, you mentioned many mutations in the this GCC. So did you also find any mutation associated with any clinical phenotype in the associated phosphodiesterase?
We haven't looked. Sorry, Shailaja, we haven't looked. I mean, even figuring out variants in one gene is a lot of effort. But when we get the whole exome sequencing from our IBD patients, pediatric IBD patients, I will specifically look for co-occurrence of variants in other genes in the GCC signaling pathway, whether it's PDE5, PKG2, CFTR, NHE3, because they may be more subtle changes. I'm not a very firm believer of a monogenic cause. I mean, there are a few, of course, but I think it's a mutation in the context of other variants that result in the severity of the phenotype that you see. Now, GCC is a driver, no doubt, but if you have a PDE5 mutation or a PKG2 mutation, along with the GCC mutation, maybe the symptoms are far more severe or less, as the case may be. So. But we will look at that when we get our whole exome.
Yeah, because that will be interesting that other than GCC function, whether PD is also playing a role. Secondly, I was also thinking, what is your take on the differential microbiome in the patients?
Yeah. So, in the microbiome analysis that they did, they took the fecal microbiomes from the patients and compared it to a so-called normal microbiome of thousands of patients in a central repository in Oslo. So it is not the microbiome of the unaffected child, for example, right? Because if you look at the family, there are people who are normal in that family as well.
Yeah.
So they didn't do that. Yeah. Now, the way we are trying to address that question is by doing classical approaches of co-housing of mice. So we can take wild type and S8 mice. Are we able to switch the microbiome just by co-housing them? Or is it something that's inherent to the environment of the gut in S8 mice? So there were a few changes that they observed in the microbiome studies in Norway, but I can't say that they were done the way a classical microbiome analysis should be done today, where you have same family, unaffected, affected individuals, and those kinds of things. Yeah.
Okay, thank you.
Thanks, Nishit.
Yeah. Babatosh also had a question. Sandhya. Babatosh, are you there?
Nice talk, ma'am. So I have just two questions. The first question that when you think that microbiome could be one of the potential reasons for the disease severity, so have you checked the effect of antibiotic in the disease severity in your mutant populations?
We're doing it now. We're treating these mice with antibiotics, and then we propose to do a DSS treatment to see whether the DSS effect is inherent or is it contributed by also the changed microbiome. So we're going to do that. We're starting it. We haven't done it yet, but we're going to do it.
And my second question, what is the prevalence of this GCC mutation in the IBD populations?
I think I showed you that data from Toronto. Of the patients which had monogenic causes, as reported in literature, 6% of them had mutations in GCC. But there were a large number of other mutations. One of them was also STAT1. I don't know if you saw it in that pie chart that I showed you, but STAT and many of these inflammatory genes are obviously, variants of them will cause IBD and Crohn's. It's a rare disease. I don't think it's, very common in IBD or Crohn's. So you could classify it as a rare disease, but it is there. And that's what we're interested in studying.
Okay. Thank you, ma'am.
Thank you.
Yeah, Krish had another question. Krish?
I just couldn't resist asking this question, Sandhya. So probably you partly answered this based on the question of Nishit and Baptush. I was wondering whether they ever tried in these patients or in your model, have you tried fecal transplantation?
Not fecal transplantation. When we saw lactobacillus levels lower in these mice, we actually tried to do, we fed the mice with lactobacillus exogenously in the drinking water. And we tried to look and see, number one, whether lactobacillus colonized in the gut at all. And it didn't. So the gut environment doesn't allow the colonization, at least of lactobacillus, but we haven't done total fecal transplant. We took the approach of co-housing the mice from weaning onwards. Or even co-housing the mice at the time of birth. With a wild type mother, we co-housed wild type pups as well as S8 pups. And we want to now check and see how the microbiome of both of these is affected, whether they're co-housed with a wild type mother or an S8 mother. So for example, if we take wild type pups and put it with a mutant mother, does the microbiome shift? So we're looking into all that at this point of time. But given the altered gut environment, I'm not very sure whether we'll be very successful in a probiotic therapy for these mice. It's inherent. Something's happening in the intestinal epithelium.
Because shouldn't that be happening even in the IBDs? And there is some literature to show that the fecal transplantation actually is successful.
So, this has been successful quite often in adult IBD. Now, adult IBD is also, one of the factors that causes adult IBD is the environment, right? Whether you smoke, the kind of food you eat, et cetera, et cetera. So maybe they can be corrected because there's no underlying genetic problem. It's just the environment that's made your gut go a little crazy. But in terms of childhood IBD, in predisposed IBD or pediatric IBD, I think there are a few studies where they've tried fecal transplantation, but I'm not sure whether those things would work very well, at least in this model. But we'll do it. We'll try, definitely.
Thank you once again.
Thank you.
Yeah, let me see the chat box something. Yeah.
Sure.
Can I ask?
Yeah, Guru has a question. Guru?
Yeah, one last question, Sandhya. Have you looked at zinc levels at all? Is there any report connecting cyclic GMP with zinc?
We know that GCC is inhibited by zinc, chloride, zinc sulfate, okay? Again, preliminary data not published. When we've looked at the IC50 for zinc, we see that the mutant receptor is more sensitive to zinc than the wild type. Now, this makes it very interesting because that means that maybe these mice can be treated with lower doses of zinc and you might still see an effect if all effects are mediated by cyclic G production. We've treated these mice with zinc sulfate in drinking water. As I said, the fecal water content reduces, the bowel frequency reduces, the Paneth cells seem to recover their function, but it's iffy as to whether the STAT signature and the ISG signature comes down. I think we need to do a few more experiments to either say it doesn't or it does selectively. So zinc may help the diarrheal phenotype, but may not help the inflammatory phenotype.
You haven't measured cyclic GMP levels under those conditions?
Yes, we have. In zinc-treated mice, cyclic GMP levels come down.
Come down.
Come down. Come down.
Okay. Thank you.
Shinjini?
And there are other markers as well, like soluble.
Shinjini? You have to unmute.
Sorry. Sandhya, looking at zinc, there is a lot of real life experience on how zinc does bring down, and it's part of the national policy, as we all know, for treatment of diarrhea. Have you, so the one question was what Guru already asked about looking at zinc and cyclic GMP levels. Have you done any of this in children with diarrhea with your clinical partners?
No, not yet, but we propose to.
That would be interesting to see. I mean, even though it will just be an association, but it will
exactly. Yes, absolutely. Exactly. In our mouse model, we see a reduction in cyclic G. But we might also see a reduction in cyclic A. We may see that too. It doesn't negate the effect of zinc.
Yeah, Sandhya, if I can ask a couple of more questions. Yeah.
Sure, Ramadi, pleasure.
Have you verified your immune signatures in clinical samples?
We haven't.
Okay.
We will. But there's a wealth of information out there in terms of the immune signatures in Crohn's disease and IBD patients. I mean, there's a lot of information. And interestingly, my student tells me, Avipsa tells me that actually the changes that we see in our immune signature are those that have been reported in IBD and CD patients. So there is something going on there, but it looks that it might, again, correlate.
So based on your talk today, it seems that STAT1 signaling is the main player which is leading to this resolution, right?
Yes, yes.
So do you think that STAT1 inhibitors can be also an alternative therapeutic approach?
Yes. So actually, we thought we would try JAK inhibitors, STAT inhibitors. It would be absolutely great to be able to specifically knock down STAT in the epithelial cells in our transgenic mouse. There is a villain STAT knockdown mouse available, and you know, but you also understand the difficulties it is to do animal breeding and animal husbandry in India at this point of time. And I'm up to here with what we have, but absolutely, it would be something that would be definitely worth looking at.
And what is your take on maybe finding activators for phosphodiesterase 5 and then use them as therapeutic?
So you know, the one activator of PDE5 is cyclic GMP itself.
Oh.
So this is what Shailaja worked on. We have the GAF domain in this PDE5 where cyclic GMP binds. And when cyclic GMP binds, it activates the phosphodiesterase. So it's a sort of feed-forward kind of, or feedback kind of thing that's happening. Other than that, even though PDE5 is so important, at least in the terms of sildenafil citrate and Viagra, there is no crystal structure published yet. I'm 100% sure Pfizer and a lot of these companies are sitting with the structure, and they must be looking to answer questions exactly as you're saying. But they may be looking more at the level of inhibitors rather than activators. But cyclic GMP is a known activator, which now results in phosphorylation of PDE5, which now results in its increased therapeutic activity. So there's a kind of...
And is anyone trying to solve the structures of guanylate cyclase with all these mutations? Or?
I have a collaborator, very preliminary stuff he's been trying to do. He's located in France right now. We have sent him all our constructs, and he's hoping to try to get some structural information shortly. Of the full length receptor through cryo and the individual domains as well, the fine kinase homology domain, etc. But as I said, there is no structure available. And the predicted structures, even using AlphaFold, don't tell us much in terms of the effects of the mutations as we see them. So, you know, it's we need the structure.
So there's a question in the chat box by Dr. Ruchi Tandon. she's asking, by any chance, did you look at PD4, as PD4 inhibitors are also being currently evaluated for IBD?
We didn't look at PDE4 because PDE4 is the cyclic AMP specific phosphodiesterase. And as I said, our focus has been on cyclic G. But it's quite likely that, I have to think, but I don't think we saw changes in cyclic AMP levels in our transgenic mice. I have to check with my student whether we've done it, but I don't think cyclic A is the driving force here. It's cyclic G, and PDE4 will maybe have a different effect depending on another pathway that's activated. It may not regulate pathways which depend on cyclic GMP.
Yeah, I don't see any more hands raised, nor any question in the chat box. Yeah, so if there are no more questions, I would like to thank you, Sandhya, for kindly accepting our invitation and giving this excellent talk today.
It's been a pleasure.
Special thanks to everyone who has joined there and interacted with you. I would also like to thank Grishma for coordinating this THSTI Distinguished Seminar Series. And I would again request you to visit us during your next trip to Delhi. It will be really nice to have you at THST and interact with you. You can bring in your mice for Guru to do his.
I talk to Guru first.
Yeah. And last but not the least, we have a memento which we could not give it to you, but we will get it delivered to your lab soon.
Thank you so much. Thank you.
And thanks again for finding time and delivering.
It's been a pleasure.
And we look forward to seeing you soon. Thanks a lot.
It's been a real pleasure. Thank you all for your excellent questions. It made me think more, which is what this is for. Thank you all so much.
Thank you very much.
Thanks a lot. Bye-bye.
Thank you.
Bye.
Thank you.
Bye. Bye.